I want us to look at the nucleic acids today. Basically, we have got two nucleic acids. One is DNA, another one is RNA. So you must also know that nucleic acids are made of nucleotides. And when you look at DNA, usually DNA it is found within the genes or on the chromosomes. That's why you find DNA, and it is also found in mitochondria and in the chloroplasts. Chloroplasts are found within plants, and this is very important because examiners like it. They always ask, where do we find DNA? So you must also know how to describe the structure of DNA as per Watson-Crick, and you also look at the structure of DNA. You must know it excuse me, to know that DNA is the natural structure which we call a double helix, and to know that DNA is made up of two strands. It is made up of two strands, you must know it, which is different from RNA because RNA is made up of single strand. So when you look at the components of DNA nucleotides, I mentioned up that nucleotides are the building blocks of DNA, or what makes up the DNA. So you have got what we call nitrogenous bases, which are linked by weak hydrogen bonds. Those hydrogen bonds must be weak and in a position to break during protein synthesis. So those bases in DNA, we have thymine, which is T. You have adenine, and is represented by A. You have cytosine, C, and you have got guanine, G. And you must know that base pairing in DNA will always follow AT and GC. What do you mean by AT? Meaning that A can join a T and C can join C. They can be able to join, and you must know that the DNA has also a sugar portion which we call deoxyribose sugar in DNA, and a phosphate. So briefly, what makes up a DNA are nucleotides. You have got the sugar component, which is deoxyribose sugar, and you have got a phosphate part. Portion means part. Then under DNA also you must know the structure of DNA, and the functions of DNA are very important. One, you must know that DNA makes up genes which carry hereditary information, and then you must also know that DNA contains coded information that is responsible for protein synthesis. Then you look at DNA replication or duplication. This one takes place, of course, within the DNA. Of course, the process happens as follows. You must know that when the cell cycle takes place, and where the cell, where the cell unit takes place, and you must also be able to know how DNA replication or duplication takes place, and this happens under the influence of the enzyme, but usually we don't go deep according to your level to describe which enzyme and how. Then you must also know the significance of DNA replication, significance or the advantages of DNA replication. Then you must also know what we call DNA profiling. Like, for example, when you go to Home Affairs, where they take your fingerprints and have the database for your fingerprints, what is the advantage of that? So you must know how to interpret the DNA profiles. You must know the DNA uses in the profile. Then also it is very important for you to take note that the other part of nucleic acid is RNA. And as I said, that RNA is single-stranded. So one, you must know its location. So where is it located? It is located on messenger RNA, and also you have got transfer RNA. These are basically two, and you also have got ribosomal RNA, which is very important. So you look at the the structure of RNA. As I said, it is single-stranded, and when you look at the components of RNA, still you have nitrogenous bases, you have the sugar part, and you have the phosphate group. The difference here between RNA and DNA also is that thymine will change to uracil, which is U, meaning that you have got adenine, cytosine, and guanine. So the base pairing stays the same. It stays the same, so you have got to look at the structure of messenger RNA. It's very important, you must look at it. You look at the functions of RNA. What role does RNA play in protein synthesis? So when you look at protein synthesis, simply means the making of proteins either in plants or in animals. So here is the process. We are saying that basically it takes two parts. You have got transcription and translation. So during transcription, you must know that DNA double helix opens up or unwinds up, and DNA double-stranded structure will unzip, meaning the weak hydrogen bonds which are holding the two strands of DNA will have to break, and you'll have one strand coming out as a template, which we call messenger RNA, using the RNA nucleotides from the nucleoplasm. And then you also have got the messenger RNA will be complementary to DNA. So you must also know the word complementary, meaning that what you have on RNA must be able to fit in or onto what you have on DNA. And the messenger RNA now will have to be coded the message for the protein. It will have to carry the message of the protein, meaning what type of the protein that is going to be formed. Then messenger RNA will move from the nucleus to the cytoplasm and attaches itself onto the ribosomes. So to attach itself on the ribosomes, it is helped by the ribosomal RNA, meaning that that's the process of transcription. So in examination, if you are asked to describe what transcription is, basically that's what is needed. Then translation involves the interpretation of what the messenger information has brought in, meaning that each transfer RNA will carry specific amino acids, and when the anticodon on transfer RNA matches the codon of messenger RNA, then the transfer RNA will bring the required amino acids to the ribosomes, and amino acids will become attached to each other by the peptide bonds to form the required protein. So there are new words here which you need to understand: the codons and the anticodons. Now, codons are the nitrogenous bases you find on messenger RNA, and anticodons are the nitrogenous bases that you find on transfer RNA. All right. So in the next video, I will talk about meiosis, and I will also have to give you a variety of past paper examinations questions on how the nucleic acids are being assessed. so that you can get exposed. Thank you.